Okay, got it.
Yeah, there we go.
Okay.
So sorry. Okay, so we are live. I'm gonna give just, I know we're a little bit late, but I'm gonna give at least like a minute or two for people to come in and realize that we're actually live now. Okay, so I will start with the introduction. So hello, everyone. Sorry for the technical difficulties. Welcome to the second live stream event from the Paper Trail. My name is Bria Macklin, and I am currently a postdoc at the Gladstone Institutes. So the Paper Trail is an online interactive journal club that was developed by four former colleagues turned lifelong friends. So myself, Dr. Michael Blatchley, who's currently a postdoctoral fellow at CU Boulder, Franklin Hall, who's currently a graduate student at Johns Hopkins University, and Dr. Quentin Smith, who is an assistant professor at UC Irvine. And Quentin is going to be your host today. But just a little bit of more background on the Paper Trail. So born from our love of discussing science and our yearly Paper a Day challenge, we came up with the idea for the Paper Trail to be a platform that allows the primary authors of recently published works to come and share the story behind their research. And so we really wanted the Paper Trail to stand out from other online presentations. And so to do this, we came up with a set of guiding questions that prompt the author to share more than just the results of their paper. And so we're so happy that you decided to join us for this live stream. And so in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. So Quentin's going to be your host, and the other hosts will serve as moderators, and we'll be in the chat. And so we'll be able to relay your questions. And Michael is eager to answer your questions throughout the presentation. So please don't hold them to the end, ask them as they kind of pop into your head. And so if the chat feature is not available for you right now, what you can do is create a Twitch login. It just takes a few seconds, and then that will give you access to the chat feature. And so saying all that, I would like to introduce our speaker, Dr. Michael Palmer. So Michael received his bachelor's degree in chemical engineering from the University of Delaware and his PhD from Princeton University in the Department of Chemical and Biological Engineering. He is now a postdoctoral scientist at the Marine Biological Laboratory in Woods Hole in the Gillis Lab. His research interests are centered around using insight gained from studying unique biology to improve engineering technologies. So past experience in this area ranges from using methylotropic genes for metabolic engineering and biofuel production, studying the development of diverse lung architectures to pursue tissue engineering applications, and now studying cartilage development and regeneration in fish for potential applications in stem cell therapeutics. So I will let Quentin and Michael take it away. Michael, you can start sharing your slides now.
Okay, awesome. So I'm assuming everything's showing up okay. But yeah, so thanks so much to the organizers for inviting me to speak today. This is a really cool idea for our journal club, and I'm really excited to share some stuff about our paper. So that paper is called Stress Ball Morphogenesis, How the Lizard Builds Its Lung. And it was the result of some really great collaborations with a lot of really awesome people. So there are so many people that I have 2 acknowledgement slides, so bear with me while I go through them. But first of all, Brian Nerger was our 3D printing and material science guru. Katie Goodwin is an expert on bioinformatics and helped a ton with single cell RNA sequencing and downstream analysis of that. Both of those people were grad students in the Nelson Lab. Katie still is, but Brian has since moved on to a postdoc. Anvita is a graduate student in mechanical engineering in the Koshmer Lab. and she did a lot of the computational modeling that ended up in the paper. Sandra is or was a postdoc in the Nelson and Tocher labs, and she worked on the optogenetic component of the paper, as well as imaging the biosynthetic model that we used for our in vitro system. Pav was a postbac in the Tocher lab. He's since moved on to grad school, but he also worked really heavily on the optogenetic cell development. Sarah Paramore helped a ton with imaging the adult lung tissues. She's an expert in imaging big organs like that, so she was really great. And then, of course, we had some really great PIs who helped with sort of formulating everything and just coming up with great ideas and being really inspiring people. So Jared Tocher, Andre Koshmerl, and my PhD advisor, Celeste Nelson, were really awesome throughout the entire process. Yeah, so that's our team, and people worked on different aspects, but it was really a collaborative effort, and so I'm excited to share the resulting.
So Michael,
yeah,
I just have a question. Was this like a natural progression? Did everyone just kind of come together, or how did this like team assemble? Because it seems like a lot of key players, from bioprinting to like optogenetics. Like how do you actually get that team together?
Yeah, so that's a great question. It took a while to get the team together. I'll talk about the timeline a little later, but the project started off rather small. So we were just sort of investigating the lizard lung and sort of curious about discovering, principles of development there. And then after we found what we found in the development there, we thought that it would be really great to pull in some other things and really tackle this problem from a few different angles. And so that's sort of how the collaborations formed. But yeah, it was sort of a stepwise process. So yeah, but I'll talk about that a little bit later with the timeline more too. Yeah. So sort of some background and motivation for the project. So here's sort of a schematic of a mammalian lung, human lung specifically. And so this is what we use. Everybody breathes about 20,000 times every day, which is quite remarkable. And so when you breathe in a mammalian lung, air flows out to these terminally branched structures, which are coated in this vasculature, and that's how you exchange gas with your blood. But this is not the only lung architecture that exists in nature. So birds, for example, have a super different lung structure wherein air flows in a continuous loop. And so it's not terminally branched like the mammalian lung. And then lizards, on the other hand, have a super simple lung in most cases. So in a lot of lizards, you have sort of this just simple balloon structure where air flows around and there's vasculature that sits along the surface of this, but really no intense branching to speak of. And so having observed just the fact that there is such a diversity in lung structures sort of motivated this project to begin. We were curious if developmental mechanisms are conserved across some of these species, and if there's anything we can learn by studying non-mammalian systems. And so, as I mentioned, the adult lizard lung is a single corrugated chamber. And so here's just a Z-stack through a lung stained with ecadherin. And here's that same, or one of these lungs projected in 3 dimensions here, just so you can sort of see how empty it is. So basically, the lizard lung is just effectively a hollow balloon with these like really simple corrugations along its surface to increase surface area for gas exchange slightly, but really no intense branching to speak of, and certainly nothing on the complexity level that we find in mammals or birds. So any questions about, yeah, sorry.
Yeah, I have a question. So, as tissue, I'm a tissue engineer myself, and I'm kind of really like focused on this idea of like biomimicry. Like we need to recreate like the human lung. So why look at the lizard? What's the emphasis of the lizard, and how did you get these images? Like how did you access these lizard lungs?
Yeah, so these are lungs that are fixed and stained. So and then yeah, just imaged on a nice confocal microscope. But yeah, sort of the inspiration. So as you say, there is sort of this emphasis in the field focusing on mammalian development and trying to recapitulate mammalian development exactly. But that proves to be rather difficult. So as I mentioned, the mammalian lung is extraordinarily complex compared to the lizard lung. And so the goal here was just sort of to see if we can boil some principles of lung development down to basic components. So if we can study a simple lung, yeah, I have a slide about this later, but basically, yeah, so if we can learn something universal by studying a lung that is so simple, basically understanding the mechanics on a basic level. Yeah, and so that's sort of the justification. And what we'll see later is that it ends up teaching us some interesting stuff about how we can use cells to pattern materials and tissues. So sort of another element of background here is a discovery that came from our lab a while ago. And so that is that this tissue called smooth muscle is important for mammalian lung development. So all lungs consist of this epithelium, shown here in this black line. It's surrounded by mesenchymal cells. And from that mesenchyme, these cells called smooth muscle differentiate. And so as they do so in the mammalian lung, they wrap around the epithelium and they actually exert a physical force on that epithelium and sculpt it. And so what our lab discovered a few years ago was that smooth muscle is incredibly important for proper lung development in mice. And that over time, that mechanism gives rise to this incredibly complex lung structure that allows mice and all other mammals to breathe the way that we do. And so
Sorry,
sure.
that actually brings us to a question from the chat. So thank you so much to our Twitch followers. We have a question. So they ask, are there some species that bridge the gap between the different lung morphologies? Or is it very cut and clean that every lung kind of goes under its own distinct form? So is there like a certain animal that bridges this gap, or?
Yeah, so there definitely are animals that are sort of in between. So for example, alligators and crocodiles, which are quite closely related to birds, have a more bird-like lung when compared to other reptiles. So there definitely are some animals that are sort of in between. And one of the exciting things that the Nelson Lab is working on, unfortunately, I'm no longer a part of it, but moving forward, looking at some other lungs that are sort of in between to try to see, how those evolutionary changes occurred.
Thank you. That's so cool.
Yeah, that's a great question. Okay, yeah. So what's quite amazing about the mammalian lung is that it goes from this simple wishbone structure here at E11 to this incredibly complex branch structure here at E15. And so while we know a fair bit about mammalian lung development, really nothing was known about reptilian lung development when we started this project. So here I'm just showing a few images of reptilian lung development. I'll dive into a lot more detail on those different stages as I progress the talk. But sort of when I started this project, it was a black box of development. And so sort of the two motivating questions for us here were just sort of how, if at all, could we use this to study tissue engineering. Let me see if I can move this. There we go. I just wanted to get the meeting controls out of the way so I could see everything. Yeah, so if we could use, as Quentin mentioned, sort of these basic principles of lizard lung development for tissue engineering applications. So as I mentioned, the lizard lung epithelium is really simple in its structure. It begins as basically this smooth epithelium, and the extent of branching that occurs is just this rudimentary corrugation along the surface. And so by studying that, could we learn about how to sculpt other simple tissues by studying this system? So getting started. What people may be surprised to know is that even picking a model system for this project was not super cut and dry. So historically for reptilian development in anoles, people use the green anole. So it has a sequence genome. Historically, it's been used But what we found is that it's very sensitive to environmental conditions. So it ended up proving to be rather difficult to keep these guys in captivity and have them breed successfully. And so after about a year of trying to get this to work, we actually switched model organisms. So we ended up using the brown anole for this study. And so the genome is similar enough to the green anole to be useful. So we can still use all of the techniques there. It's an emerging model system that's quickly gaining traction for the reason that it is an invasive species. And so it's resilient to changes in environmental conditions, and it's super easy to keep in captivity. So they're happy and they breed for a large portion of the year. And so that proved to be really great for this project. And so sort of a timeline of how this project ended up taking shape. So in July 2017, the first animals arrived on campus. So those were the green anoles. And for basically a full year, we tried to get those guys to work for us, and they didn't. So in June, we started with the brown anoles. And so then the next stage of the project was studying development. And so we studied just the lung development, and we were curious about all those developmental mechanisms. And then in July 2020 was the first meeting between all the collaborators, which, as I put this on the timeline, I had to double check was correct because that seems very recent. But it really was a whirlwind, and this project came together very quickly. So it took about a year from when we first met to when the paper was submitted. So April 2021 was when the paper was first submitted, and then it was published in December 2021. And so, sorry, go ahead.
That's a very a long timeline. I think it's good to kind of highlight, how long it actually takes to develop these systems. Was it difficult to get lizards on campus? I know there's a lot of mouse colonies and other things that are kind of in place. Were there any special considerations for these invasive species lizards? What does that process look like?
Yeah, so I mean, all research organizations have similar animal, the IACUC has to approve everything. And so we had to submit new proposals and get the rooms set up. Basically, the anoles were a new system for Princeton entirely. And so we had to really work to getting the systems up and running and to getting rooms that were satisfactory for keeping animals like that. So it was really a process. In terms of getting them up here, we got some shipped up here, and we also ended up going down to Florida to collect some of our own, which was really exciting. I think I'm the only chemical engineering grad student to have done field work like that, so it was pretty fun. But yeah, it was quite a long process, and it took a lot of trial and error. A lot of people who do reptilian work do it in the southern US because they have such access to the animals. They live there natively. So doing this work in New Jersey definitely had its challenges.
So did you have to catch them, trap these lizards? How did you
Yeah, so you do catch them with a little lasso, basically made out of dental floss. It's quite an adventure. It's super fun though.
Oh, that is awesome.
It is great. Yeah, very cool. So yeah, that's the timeline. And I'm sort of going to break the talk up into two sort of phases of this paper. So the development phase of studying lung development, and then the big collaboration phase. So that's the biosynthetic model in our in vitro system, as well as the computational modeling portion. So to sort of break down lizard lung development even further, there's sort of a couple different phases here. And so the first phase that I want to talk about is this inflation stage. So the lizard lung starts off in this simple wishbone shape, looks super similar to the way a mouse lung would start or a chicken lung would start. But then rather than branch right away, as occurs in the mouse and birds, the lung inflates. And so as it does this, you get this incredibly dramatic thinning of the epithelial cells here. And so the lung is really acting like a balloon and inflating there. And that's observed here with the super dramatic thinning of the epithelium. And then after that occurs, you get the formation of these corrugations along the surface of this inflated lung. And one of the first things that we were curious to examine, of course, is whether smooth muscle might be important here, because we know that it's important in mammalian lung development. What we found is that initially smooth muscle differentiates pretty diffusely in the mesenchyme, but over time it refines itself into these really beautiful bundles. And this really is a muscular tissue. It's contractile. And so when you take lungs out of the embryo like this, the smooth muscle continues to contract. And it looks a little freaky, but it's super interesting. And so here you sort of get an appreciation for the types of forces that this muscle is able to create on that relatively soft underlying epithelium. And so having observed this phenomenon that the epithelium only forms these corrugations in the gaps between this smooth muscle mesh, we are naturally curious, is smooth muscle essential for corrugation formation?
So Michael, was this expected? Were you guys surprised about like the architecture of these kind of stromal populations with the epithelial cells? Was this like surprising as you do this? And like, I'm very intrigued by this image. Is this like an explant that you embedded in a gel, or like, how did you get these images? They're beautiful.
Thank you. Yeah, so the movie is just a lung that's sitting on like a little membrane that's floating on culture media. So enough media diffuses through the membrane to keep the lung, with enough nutrients. And it's exposed to air as well, so it gets all the oxygen it needs. And so those, I'll talk a lot more about the sort of culturing experiments that we can do later on, but lungs like this are plenty happy on a membrane like that for up to 48 hours and even beyond. So that was really nice. It was nice to have a system like that worked well for culturing. The images on the right are just collected. These are fixed and stained embryonic lungs at different stages. Yeah, I mean, we're lucky because these tissues are really small and they're relatively clear. And so imaging them produces these nice crisp results. So I often say that I was very lucky to work in a tissue that's this nice without me having to do much to it. But yeah.
Fantastic. We're getting a little bit of audio feedback. Is there a way we can readjust that?
Okay. Yeah, let's see. I wonder if...
Yeah, it's just like a little echo.
That's strange. I guess I could try without the headset.
Okay.
Is that better?
Still a little bit, but yeah, that's good. Thank you.
Okay. All right. We'll try it like this. Yeah. Sorry about that.
Okay. No worries.
Okay. But yeah, so having observed here that this smooth muscle forms these like really nice bundles and these corrugations protrude, and the gaps between that smooth muscle, we were curious, is smooth muscle essential for the formation of those corrugations? And so in order to investigate this process directly, we needed to know how that smooth muscle was showing up in the first place. And so we did single cell RNA-seq analysis here to sort of probe the mesenchymal population to get a better understanding of how they were feeding into that smooth muscle population. And so what we observed here is that along the differentiation trajectory of smooth muscle, we see an increase in genes associated with the sonic hedgehog signaling pathway. And this was really exciting to see because this is also true in mice. And so this was sort of like a nice confirmation that, this system does behave somewhat similarly, albeit in a very different pattern. And so armed now with this knowledge, we were able to investigate smooth muscle directly in our system. And so we treated lungs, explant cultured lungs with cyclopamine to inhibit sonic hedgehog signaling and decrease smooth muscle differentiation, and with smooth antagonists to promote sonic hedgehog signaling and smooth muscle differentiation. But what we found is that when we get rid of sonic hedgehog signaling, we don't really get any smooth muscle differentiating from the surrounding mesenchyme. And when we promote sonic hedgehog signaling, we end up with all this excess smooth muscle. And what you'll notice is that in both cases, this gets in the way of branching. And so it seems like there's really this optimal amount of smooth muscle that differentiates, that sort of allows for it to reorganize into these bundles and effectively undergo development. But basically, the takeaway from this set of experiments is that smooth muscle is incredibly important for the formation of these corrugations. And when you get rid of it, you don't get those forming. So
Michael, I really like that experiment. So just in general, when you're doing these like single cell sequencing approaches, a lot of things pop out, a lot of things kind of signal. So how did you choose, sonic hedgehog? And are there other, potential mechanisms that kind of drive this patterning of the stromal cells during lung development?
Yeah, so there's definitely lots of things that can factor into it. So one of the first things we did was start by looking for known signaling pathways in the mouse. So people are sort of quick to assume that like these types of experiments are like fishing expeditions or whatever.
Yeah.
But so it does help to go in sort of with an idea of what you hope to find, and so that way if Sonic Hedgehog was important, that's a cool result, and if it wasn't, then that's a cool result too, because it suggests a divergence in mechanisms between these systems. So yeah, that's how we settled on the Sonic Hedgehog, was sort of a comparative approach there. And so having observed then that smooth muscle is important for development, we wanted to examine that smooth muscle on a much finer level. And so we started off just imaging its development on a much higher quality. And so what I mentioned before is that smooth muscle initially begins sort of diffusely patterned in the mesenchyme. And at this stage, these smooth muscle fibers lack any sort of intense orientation. They're sort of just all chaotically aligned there. But as development continues, those become really aligned. And so what you'll notice here is that the smooth muscle fibers are sort of oriented circumferentially around the lung. And then over time, those will refine themselves into these initially horizontal fiber bundles. And then as development progresses, those will become connected by these sort of pseudo-vertical bundles, eventually resulting in that fully formed meshwork. And so there's sort of two phases of smooth muscle bundle formation. There's sort of this initial horizontal bundle alignment, and then later on you get these connective bundles that complete the mesh. And so we were naturally curious, how do those fiber bundles form? One of our first thoughts was perhaps because we go from such diffuse smooth muscle patterning to a more refined one, maybe there's some sort of patterned apoptosis of those smooth muscle cells. And so we examined that directly, and that proved not to be the case. So cross that one off. And then we were curious about mechanical forces. So I mentioned earlier on that the lung at these early stages undergoes this really intense inflation process. And so because that's probably a very mechanical process, we hypothesized that maybe that could be important for patterning these smooth muscle bundles. And so that's what we dove into. So something that we observed is that the lung aspect ratio changes throughout bundle formation. So you have sort of these three distinct stages of lung development here, each with their own like unique characteristic aspect ratio. So of course, in this wishbone phase of early lung development, you have a rather small aspect ratio of the lung. And then while the lung is inflating, it has quite a high aspect ratio. And then interestingly, so that's when the horizontal bundles form. But then interestingly, when you get these connective bundles showing up, you actually see a decrease in the aspect ratio again. And so that suggests to us that the lung is undergoing various phases of tension, and we wanted to examine that directly.
So Michael.
Yep.
Sorry for interrupting, but we're engineers, so I'm like really intrigued about like what tools that you like. Why did you choose to quantify these particular morphological changes and kind of this aspect ratio in the way that you did? And there's a lot of quantification in the paper, and I don't want to ruin it, but like as an engineer, was this kind of hard for you to like kind of develop these systems? Like this seems like a very biologically driven problem, but can you just like highlight a little bit of like the engineering lens that you used in kind of tackling how does the lizard form its lung?
Yeah, definitely. Yeah, so I mean, as you said, I have an engineering background, and so mechanics and forces are always playing on my mind. And so sort of as soon as I observed that the lung was inflating like that and the epithelium was thinning so dramatically, likely we assumed, and I'll show the results later, but downstream of some internal pressure, my brain started going like, there's got to be some forces here at play. Like this feels like a very mechanical system. And so that's how we sort of started, thinking, well, how can we show these things? And so the first thing we did here was this aspect ratio quantification, just showing that there are these different phases of the lung development. And then the next slide I'll show you is sort of perturbing those internal forces directly and seeing what happens. So, yeah. So, as I mentioned, this lung inflates. There's pressure, or there's luminal fluid being secreted into the lumen and increasing the pressure. And so one of the first experiments that we did here just to see what was going on was we punctured these lungs. And so when you pop them out of the embryo and you puncture them, they deflate. And so if we quantify strain here in the two different directions, so sort of horizontally here, which is also circumferentially or vertically, what you'll notice is that the strain along the horizontal direction is quite a bit more than we see in the vertical direction. And so that suggests to us that at this inflation stage of development, there is some oriented tension and it's circumferential. And so there's a clear interplay sort of between this internal pressure caused by secretion of fluid into the lumen and the tension experienced by the lung tissue. And so we observed here that by effectively popping the lungs, we could observe that those forces in real time.
I love this experiment. I love the engineering aspect. This is great.
Thank you. Yeah, it's sort of like a brute force way of getting at it, but it works nicely to illustrate everything. So thank you. Yeah. And so now knowing that, or now highly suspecting that the internal pressure might have something to do with this tension, and as well as the downstream formation of those smooth muscle bundles, we wanted to affect that luminal pressure in a more serious way. And so there were a variety of ways that we could test this. And so what I'm going to show here is control lungs. We punctured these lungs with these hollow glass needles. And so the needles stay in over 48 hours. And so that prevents any wound healing mechanism from closing the lung back up. So basically this prevents pressure from accumulating within the lung. And we also treat it with a drug called forskolin, which just promotes secretion of fluid into that lumen and so increases the pressure there. And these treatments do what we expect them to do. In a punctured lung, you end up with a smaller final lung. In a forskolin treated lung, it's bigger. But what we observed sort of in downstream of that in terms of the smooth muscle was really quite exciting. And so namely that in control lungs, you end up with these nice fiber bundles forming. But when we puncture the lungs or we increase the pressure too much, then you end up with sort of this retention of like a chaotic smooth muscle pattern. And so in both of those treatment cases, smooth muscle fails to reorganize itself into those bundles. And also as a result, because they don't form those bundles, you don't end up with those corrugations that you see during normal development. And so what this suggests is that during development here, there's sort of this ideal pressure that the lungs achieve, where it's enough to sort of guide those muscle cells to reorganize themselves, but it's not so much that it's overwhelming, as is the case with forskolin. And so having observed that and sort of realized that tension downstream of luminal pressure is responsible for the formation of those horizontal bundles that form first, we started being curious about the second phase here that I talked about. So when the lung goes from this inflated stage with a relatively high aspect ratio back to something with a slightly smaller aspect ratio. And so we were curious, does the contraction of those initially patterned horizontal smooth muscle bundles result in the change in aspect ratio? And is that contraction required for the formation of those connective bundles between the horizontal bundles? And so we treated lung explants here with a drug called Nifedipine, which just inhibits smooth muscle contraction. And what we observed here is that in control lungs, you get those nice connective bundles forming. But in the Nifedipine treated lungs, you still get a few, but it's significantly less. And that also affects the aspect ratio of the lung. So when we prevent those smooth muscle bundles from contracting, you get a lung that stays sort of with that increased aspect ratio, which is more reminiscent of the inflation stage of development. And then here's just the quantification showing that those smooth muscle bundles are primarily horizontal in the Nifedipine treated lung, as opposed to being more distributed evenly in the control lungs.
That's great. We have a question from the stream. Thank you, Twitch streamer. Can you characterize like the fluid that's in the lung? Like, is the viscosity of the fluid that's in the lung, is that important for some of these like morphogenic events?
Yeah, that's a great question. So we have not characterized the fluid properties of the luminal fluid, but it would be really interesting to know sort of, what the viscosity is, what it's made up of, things like that are ongoing questions that would be cool to address. Yeah. Okay, so sort of having now observed these two phases of muscle bundle formation, the initially horizontal formation downstream of that tension from the pressure, and the formation of these connective bundles downstream of tension created by contraction of the initial horizontal bundles, we wanted to see if we could examine this real time. And so sort of a thing that we tried that we didn't know if it would work was we treated these lungs with GFP adenovirus. So we injected it directly into the mesenchyme in areas that we wanted to visualize. And so in mammalian systems, this adenovirus infects cells and expresses GFP. But to our knowledge, nobody had ever tried this in a lizard system. And so we didn't even know if it would infect. It does. And so we are very excited to see that we can label mesenchymal cells. Unfortunately, we can't label those mesenchymal cells with any specificity. But what you'll notice here is that you can quite easily see which cells are smooth muscle cells because they form these connective bundles. And so we were actually able to visualize in real time the formation of our smooth muscle bundles using this adenoviral technique. And so, of course, we were then curious, what are those cells doing in a punctured lung? So when they don't have those tension-oriented cues. And so when we puncture the lung, we see that we don't get the formation of those bundles that we see in a control lung. There's significantly less average displacement of those cells, so their mesenchymal cells are not migrating as much in the absence of that tension-driven cue. And there also isn't an orientation to their migration. So you can appreciate that some of these cells are, really migrating in the same direction, whereas in the punctured case, it's much more chaotic. And so sort of as a summary of smooth muscle development in the lung, smooth muscle is required for corrugations to form in the developing lizard lung. The formation of the meshwork happens in two phases. So the first is that horizontal bundles are aligned by tension, and that's downstream of the luminal pressure. And then those horizontal bundles contract and they cause a vertical tension to arise, and that forms those connective bundles. So I guess I'll pause there for questions. So that's sort of the end of the development portion. Now I'm going to dive into sort of the second phase of the more collaborative aspects with the in vitro system and the computational modeling.
Yeah, that sounds good. I think, again, this is like a beautiful display. Were those MATLAB plots? I have to do a shout out. Were those MATLAB plots?
They are MATLAB plots.
Okay,
yeah.
Great. So highlighting the engineering work. So yeah, if we can continue with this discussion, I think that'd be great.
Okay, awesome. Yeah. So next I wanted to sort of probe more the forces of development and sort of get into to other aspects that we can learn from the system. And so sort of one of the central questions after making all these discoveries was, is smooth muscle contraction or internal pressure more important for the sculpting of the sheet into that corrugated structure? And so you can sort of imagine a possibility where the smooth muscle contracts inwards and sort of forces the epithelium to bulge out, or the luminal pressure pushes the epithelium through the gaps in the smooth muscle. And so in order to investigate this directly, we developed a few different techniques. So namely the biosynthetic tissue engineering model and our computational models. So we wanted to see if we could sculpt materials using bio-inspired principles. And so sort of a proof of concept from the first experiments that we did here was just drop casting PDMS onto water, 3D printing a lapunite clay on top of it in sort of this hexagonal pattern. That clay dries, and as it does so, it shrinks. And we wanted to see if we could deform the PDMS in sort of this corrugated pattern, similar to what we see in the lizard lung. And
Michael, I'm so sorry. We had a
you're fine.
question. We have a question from our
Great.
streamer.
Yeah, that's awesome.
So someone asked, kind of going backwards, if you drop the pressure and then some way kind of reintroduce that pressure, can you regain that proper formation? So let's say you puncture the lung and maybe you heal it some way. Do you think you can regain that like stromal like assembly?
It's possible. It proves to be very difficult to do one and not the other. Yeah, the time, it sort of ends up being like a problem with the timing of development there. So, you need to give it enough time to experience one of the effects and then, fix the system and let it have the other effect. And in the 48 hours that we can, culture these lungs happily, it's tough to do so. I suspect that you'd be able to recover it to some extent, but it's unclear how much other things stabilize. So for example, I'll talk a little bit later, but there's definitely a phase at which, or a point at which the epithelium has sort of reorganized itself. And so it's sort of solidified in an inflative state. And so it eventually, when you puncture it, it doesn't, sort of collapse in the same way it does at those early stages. And so there's definitely things happening. And so I suspect that if you, drop the pressure and then increased it again, you'd get some results, but it definitely wouldn't be normal.
And was this like your original hypothesis? Like, were you really thinking like, okay, it has to be pressure, so we're gonna, try to perturb the system by different ways? Or did it kind of like naturally evolve? Because, with science, it's like the way the paper's written is beautiful. Was this like the logical steps moving forward, or were you like kind of trying things out in the lab to see, well, what if I do this, does it disrupt the system? So how'd you come up with, these types of approaches?
Yeah, so in terms of pressure being the culprit, that was sort of always a hypothesis, just because secretion of fluid into the lumen has been shown in other systems, and also because we observed that inflation very early on in the study, we suspected quite quickly that pressure was important. But yeah, in terms of designing the experimental approaches, that took a lot of sort of finagling. So getting good at puncturing the lungs with those hollow needles and getting them to deflate consistently took some effort, and finding the right concentration of forskolin to get a happy lung with just an increased pressure took a while too. So there was definitely a lot of optimization that went into the creation of those approaches.
Thanks. I think it's just good to clarify these things. If people, dive into science, it's good to know that, sometimes it just takes a little bit of, trial and error sometimes.
Absolutely. Yeah. So I will say that for this entire paper, we had, for most of it, a pretty clear idea of what we wanted to do. We were just limited by the biology and by the systems that we had. So a lot of it was trial and error and optimizing things to be able to actually answer the questions that we wanted to answer. Yeah, and so sometimes that's the way, and sometimes it is, and sometimes it's the opposite. But yeah, so here in our 3D printed model here, this is just sort of a purely material proof of concept, and so we were able to deform this PDMS sheet using this lapunite clay and get these corrugations on the surface. And so that was sort of all the proof that we needed to keep pursuing this. But what ended up being a critical method, and that took a little while to figure out, was getting a film system that we could both print on and then seed with cells. And so we have this 3D printer, and it did a really great job of printing these nice hexagonal patterns consistently. And so for the system I showed you on the previous slide, we were just printing on top of a PDMS film that was drop cast on water. But we needed something that we could submerge in media and get cells to sit on. And so we developed this protocol. And so this was quite a crucial step for us here. And so we UVO treat glass. We drop some dextran onto a slide, spin coat that, and then we drop our silicone elastomer on top of that and spin coat it again. And then we 3D print our collagen and matrigel scaffold on top of that. And then we submerge all of this and inactivate the silicone surface. Then we seed our cells and allow those to grow. We let them differentiate into muscle. And then finally, you can peel that membrane off of the glass. And so now you have a free floating thin film with cells patterned on top of it. And so this was exciting, even this just figuring out how to do it. But one challenge that we faced along the way was finding the right material. And so we wanted something that we could equate to an epithelium, so something that we could get thin enough and that was soft enough to basically have similar material properties to a developing epithelium. But the majority of commonly used laboratory elastomers were either too stiff or we couldn't make them into a thin enough film. And so eventually we ended up trying a bunch of different materials, but we settled on something called EcoFlex, which is an elastomer commonly used in the special effects industry and also for prosthetics. And it ended up being sort of the perfect material. It spun really thin, it was super soft, and it just worked great for us. But it did take several months trying different materials to get something that worked. So yeah, I just wanted to highlight that here, that certainly wasn't a linear process. And so having now developed a protocol for getting cells onto these films, we found a material we were happy with. We could finally start seeding cells onto these things. And so we did that. And so we printed our structure. It's a collagen matrix mixture. The cells are super happy to sit in there. And what you'll notice is that just naturally, they tend to align sort of along these printed lines here just because of confinement. So the way that they're sort of squeezed together, it forces them into alignment there. And so that was really great. We were able to get a pattern we were happy with. But then, of course, we needed those to contract. And so we developed this optogenetic system wherein we could force calcium influx into the cell and stimulate contraction. And so you'll notice here, all the flashes are sort of calcium influxes. And so the system works really nicely. And so when we seed our synthetic tissues with those cells and we let that membrane float in media, these cells are actually strong enough to contract that film. And so you'll appreciate over the course of this video that film or the structure there shrinks quite a bit downstream of these contractions. And the 3D result is shown here. So we are able to deform that film quite nicely using these cells. And so this shows us that, first of all, the material we selected was great. It was soft enough that these cells were strong enough to actually deform it. And then we were able to actually accomplish that here. So
did you just reverse engineer a lizard's lung, essentially?
Effectively, yeah.
Oh, that's so cool. I love these images. It's really awesome.
Thank you. Yeah, so sort of this set of experiments has two implications. So first of all, it was just exciting that we were able to use cells to deform a material like this. But it did also reveal to us something fundamental about the lizard biology, and that was that what you'll notice here is that this pattern is not super nice. It's not even, and also those corrugations ended up being quite a bit more shallow relative to their width than we see in the lizard lung. And so while this is an awesome system and is super exciting from a tissue engineering perspective, what it did reveal to us about the lizard lung development is that smooth muscle contraction alone is not likely responsible for the formation of those corrugations in the lung. And so in order to investigate other principles, we turned to our computational model. And so this video is super fast, but I'm showing the results here. So basically, when you have no luminal pressure, this is just what the mesh looks like. And when you increase the luminal pressure, you can see that we're able to get smooth muscle to form enough of a barrier that the epithelium is able to be forced out in the gaps there. And so this was really cool because we set all these parameters to be as close to physiologically relevant as we could, and we were able to recapitulate really nicely the development of the lizard lung using this model. And so next we wanted to test something that we were just sort of curious about, and so that is what happens if you contract, or if you sort of puncture a lung that's late in its development. And so at this stage, we hypothesized that those epithelial cells have been stretched out for long enough that they likely would have reorganized their cytoskeleton or their extracellular connections. And so in so doing, they will have stabilized. And so when you puncture the lung and you decrease that internal pressure, the smooth muscle will then be able to contract freely against no force anymore. And so the model predicted that in that case, you end up with a rather small lung, but with these elongated corrugations. And so we tested that in our system. So we took a lung much older than the ones we were using before, and we punctured them. And what we found, true to what the model predicted, was that we get these really dramatically elongated corrugations. So they're no longer really even corrugations. They look like branches. And so, not unlike what you would see in a developing mouse or bird lung. And so this was super exciting because it sort of revealed to us that it's possible to get these really different lung architectures just by changing the mechanical forces that these tissues are experiencing. And so, yeah, that was just a nice illustration of that. And so sort of a summary of our findings here. I talked a lot about these, early stages. So how we get these bundles forming. And then sort of the final piece of the puzzle was discovering that luminal pressure is sufficient to form those corrugations. And so from all these experiments together, the explant experiments, the in vitro biosynthetic model system, and the computational experiments, we were able to sort of capture this global perspective of how the lizard lung forms. And so it was really exciting just to have everything come together and to use these engineering principles to understand something about fundamentals about tissue mechanics, but also about lung development in a sort of unique system. And so just some conclusions, sort of takeaway messages, is that in nature there are often multiple solutions to the same problem. So gas exchange has been solved in a number of different ways across vertebrates. And then by studying the diversity of the natural world, we can uncover new mechanisms of development. And these discoveries can inspire new advances in engineering. And so that's sort of the whole approach to these types of projects that I think is nice to take is, while the engineering applications might not immediately be obvious, there's a lot that we can learn by studying sort of these non-classical model systems. So I guess I'll pause there for questions, and then I'll talk a little bit more about sort of the submission and publication process if people are interested in that.
I just wanted to make a quick comment, and then, it'd be nice to highlight kind of that submission process. But it's just so cool that, we can look at nature and be like, well, nature, did it a little bit easier. So maybe, we kind of talked earlier, I'm interested in rebuilding tissues. So maybe instead of doing the complex kind of budding architecture, maybe we could just do something like this and maybe get the same degree of breathing. I think this is so elegant. So thank you for sharing, the background behind that.
Thank you. Yeah, no, it's nice to take that approach, for sure. Yeah. And I think we learned quite a bit about sort of tissue mechanics fundamentals, and we developed a system that, hopefully will be useful moving forward. So, something that I thought might be cool to talk about is just a summary of the review process. So people are often curious to know, how long did it take, what changed during review, and things like that. And so in April 2021, we submitted the manuscript to Science. It went out for review, but was ultimately rejected in June. We were still excited that it went out for review, but yeah, it ended up not working out in Science. But they did offer us sort of the unique opportunity to transfer it to Science Advances. And so in so doing, some of the reviewer comments from Science submission were transferred over. And so it shortened the review process quite a bit for us. And we had comments already to work off of while we were waiting for the new reviews to come in. So that really expedited the process. We didn't have to, answer all those reviewer comments and then resubmit to a new journal. We could sort of work on those initial rounds of reviewer comments while we were waiting for more to come in from Science Advances. And so ultimately, we got our final review of comments in October, and it was the paper was published in December. So not a super long timeline here. I think we were rather fortunate with how everything worked out. I know sometimes this is not the case, but yeah, so that's how everything worked out for us. And sort of major reviewer comments. So interestingly, we got some people who were not, sure of the novelty of the work. So it has been shown in mice that during alveologenesis, there's, kind of a similar process that occurs, but this ends up being quite different than what we see in the lizard lung. And also sort of our approach went above and beyond with the tissue engineering aspects. And so addressing those comments ended up being sort of more of a question of more thoroughly addressing sort of the prior literature and explaining, why our findings were so unique and important. And then also some reviewers just wanted us to use a few alternative methods to confirm results. So specifically for the pressure experiments, people wanted a few different ways of us perturbing that. So we were able to get those addressed pretty quickly.
That is fantastic to hear. As you mentioned, the review process can be like years. So it's nice to just kind of see the transparency and how you were able to kind of adapt and, resubmit your manuscript, which I think is great. So we got some additional questions kind of back to the science, just like general, like what holds the pressure in the lung, and how does the fluid not escape through the mouth of the developing lung? And then, we'll start with that. And then we have another set of questions.
Yeah, so for these early stages of development, the lung isn't open to the surroundings like that. It's still closed off. So the lung maintains its pressure that way. And what's cool too is that when you take a lung out of the embryo and you're culturing it, initially, because you have to cut the trachea off, it's open to the environment. But because of wound healing mechanisms and how fast the embryo is able to heal, that closes off really quickly. And so it reestablishes that pressure just within a few hours.
That's awesome. I did not know that. And then the second question says that you mentioned that the lung appears to be closer to a mouse or a bird lung. Would that be a fatal change for the lizard, or could that be modified to improve oxygen exchange? So maybe from like a evolutionary perspective, right? Kind of in this divergence from like the lizards to birds, do you think, they can improve somehow with a better breathing mechanism? Or why do you think lizards have been like adapted to have this kind of maybe more efficient way of breathing? I think in the introduction, you talked about this like unilateral kind of flow between different systems. Can you kind of explain why the lizard might have like developed a stress ball lung?
Yes. So the stress ball is, sort of the most basic lung structure that we see in terrestrial vertebrates. And it's able to be so simple because a lot of lizards just have really low energy lifestyles. So if you see, a lizard in the southern US, it's usually just like sitting, and it can run a little bit, but they can't, exercise to the extent that other animals can. And so they're able to get away with these really simple lung structures. But other animals, when they developed these more complicated cardiovascular and respiratory systems, they were able to occupy niches that they couldn't before. And so that's when you see animals start to move to more harsh climates and have more energetically intensive lifestyles. So it sort of ends up being a trade-off between developmental complexity and the energy required to do that, and resource competition. So having a more complex lung allows you to just access more stuff and have a more intense life. So there's quite a bit of really interesting discussion too in the literature about atmospheric oxygen levels and how that affected sort of divergence in these tissues as well. So atmospheric oxygen has changed a ton over the course of Earth's history. And so in recent history as well, and sort of the changes in atmospheric oxygen have sort of driven some of these changes in lung structure too, as it became more favorable to have more complex lungs to extract as much oxygen out of the air as possible.
That's a really nice description. I like that. And hopefully, due to COVID, we all haven't gone to kind of like a low energy lifestyle.
Exactly, yeah.
And, kind of final questions as we wrap up. So you kind of mentioned that there are alternative methods to confirm the results based on reviewer comments. did any of the methods that they kind of propose like really improve kind of the methodologies or produce differing results?
They didn't change the results at all. So one example, sort of touching on the troubleshooting stuff that I mentioned earlier, was we have this device where you hook a lung up through the trachea to like a hollow needle, and that needle flows through like a chamber, and you're able to increase the pressure within the lung like hydrostatically by just adding more media on one side to increase the pressure. And that works really nicely for mouse lungs, but the lizard lungs proved to be pretty difficult. So initially I wanted to use that system for these, but the lizard lungs were so small that it ended up being kind of difficult to get them attached to the device. And so I ended up going with the puncturing and the forskolin. But then when people weren't super happy with the forskolin data, I had to revisit that. And so I spent some more time troubleshooting that and was eventually able to get it to work. So, I mean, it was nice in a way that I got to revisit something that I, had wanted to do originally, but just couldn't get to work. And so in that way, I mean, it, addressed the reviewer comment, but also gave me some resolution to the whole question that we could accomplish this, and also that, a purely physical mechanism for increasing pressure gives us the same result. So that was a nice confirmation. Yeah.
Great. Do you have any additional slides to share? There's one more question in the chat. Cool.
Okay. Yeah. So do you want to ask the question, or should I just wrap up?
I think it kind of coincides with this. So I'll let you kind of tackle this first, and I'll ask the question.
Okay, awesome. Yeah, so sort of just thinking about next steps. So I'm not in the Nelson lab anymore. I've moved on to my postdoc, but sort of some things that are unresolved and that I think would be pretty cool to investigate are performing our film experiments, but in a different geometry. So we were only able to get those really thin films on a plane geometry. But it would be awesome to get something, spherical or at least hemispherical, to be able to examine whether geometry is important at all for how much or how deep those corrugations can be as a result of smooth muscle contraction alone. So I mean, this proves to be technically challenging just because it's very tough to get an even film in a geometry like this that's thin enough for what we need. And also printing on a spherical geometry is very difficult. But if somebody wanted to devote time to that, that would be a really exciting next step for this, and have some potentially interesting implications for 3D printing like actual tissues, with a functional geometry and a lumen. So that would be cool. And then ultimately, I mean, the goal would be to replace the film with a sheet of cells. So if you could, seed a film of epithelial cells, get those to be confluent and adhering enough to each other that you could then print muscle on top of that, then that would sort of elevate the film system that we created to a more directly applicable engineered tissue system. So those are sort of just next steps that I can envision for taking the fundamentals that we developed with the tissue engineering system to the next level.
That's great. And just like kind of final questions, kind of along those themes.
Mm-hmm.
So, chemical engineer, you studied lizards. Like,
Yeah.
What other model organisms do you think bioengineers might be, missing out on? What do you think is like another cool, maybe emerging system that bioengineers should be thinking about? And then what is the next animal that you want to study?
Yeah, so that's a great question. So I will shamelessly plug my postdoc work.
Okay.
Basically, so the animal that I'm studying now is called the skate. It's a fully cartilaginous fish, and so because it's fully cartilaginous, even as an adult, it retains stable populations of cartilage cells, chondrocytes, and those are able to really effectively heal wounds in that system. Whereas in mammals, your cartilage is sort of isolated to primarily your joints. And if you injure it, it doesn't heal very well at all. And so we're going to be investigating that system to try to understand how the skate is able to maintain cartilage into its adult life and undergo that wound healing when mammals can't do it. So yeah, so that's just one example of sort of using these like unique biologies to sort of help address engineering questions. So ultimately, what we'd really love to do is sort of take what we find and try to implement those in a mammalian cell line to see if we can stabilize cartilage cells in vitro. But yeah, in terms of other systems, I don't know. I mean, there's lots of cool stuff out there. I think that historically, just because it's been easiest, people have been limited to sort of the classic model organisms. So, you know, mice, Drosophila, zebrafish, Xenopus, and those are amazing systems. But sometimes I think in those systems, you know, it can get a little saturated, or you get so bogged down in the details that switching to a new system can sort of help clear some of the, or help clear up some of the questions about fundamentals that might be hard to find in all the details of another system.
Like that. Well, thank you so much for kind of presenting this work. Dr. Macklin.
Hi, no, go for it. I think you're gonna shout out all the people that have helped you out along the way. I'm sure people would love to see this slide.
Yeah, so this is just sort of a repeat of my acknowledgement slides with some additional people who, you know, I couldn't have done this work without. So people that were instrumental in getting me started and learning how to do some of these techniques. Obviously, I want to thank Celeste again for being an amazing advisor, for allowing me to do this crazy project. And then, of course, our amazing funding sources, without which this would not be possible. And we also have some amazing collaborators, both who worked on this project and others. So we have a sort of emerging collaboration here with Paul Traynor at the Star Wars Institute, looking at developing lungs in chameleons who have sort of more complex lungs than anoles, but not quite as complex as you know, a mouse or a bird. So yeah, so that's sort of one of the avenues that we're looking in for like those transitional lungs that somebody asked about earlier. So exciting things to come, hopefully.
Thank you so much for that, Michael. I think that talk was like so exciting. I mean, reading the paper, it was really nice and it got us all really excited, but some of the videos and images that you showed just kind of really illuminated a lot of the processes and added a lot more to the story. So we're so thankful that you were able to come on and talk about your work. So for everyone on Twitch, we're going to end the stream now. Thank you so much for joining. If you want to find out more information about the paper trail, you can find our social links and our websites. I think they're somewhere below or somewhere on the screen. You can also just go to our Twitter, which is the Paper Trail JC. And we are doing this right now once a month, but we hope to add in more streams. So with more viewership and more interaction, we can do that for you guys. So like I said, thanks for joining. So I'm gonna end the stream now.